introduction. Yes, I'm going to talk today about dominance or commonest patterns in a tropical region, specifically of woody plants, mostly trees. Let's give a little bit of background for this. Traditionally, community ecology of tropical forests has focused on rarity and rare species because this is central to the question of diversity. My line of research, in contrast, focuses on the common taxa, commonest patterns, and what are the processes making some species more common than others. And the motivation, the general motivation for this line of research is applied. At the end, most of the individuals, the biomass, and the biotic interactions in tropical forests are dominated by a very limited set of species. So if we can understand common taxa and commonest patterns, that could be like a shortcut to understand the whole system. The presentation is going to have this structure. I'm going to define commonness from different angles. I'm going to explain the oligarchy hypothesis, which is perhaps the most influential idea regarding dominance patterns in tropical forests. I will present the results for two empirical studies related to the oligarchy hypothesis, and I will summarize the conclusions from these two studies. I will finish with some future directions, some open questions related to dominance. The spatial distribution is one important dimension of commonness, or one possible definition of commonness. So some species are common because they are broadly distributed in a region or are very frequent. They appear in many different sites. This is an example, Euterpe precatoria. It is a very common palm in the neotropics. And it occurs also in many of the plots that we established in our studied region too. And this is not so common a species from this point of view, which is Erythrochiton phallax, and it doesn't occur in many sites, or it's not very frequent or very widely distributed. This aspect of commonness is independent to the abundance of the species. In fact, in the Mariri region, which is my study area, this species is very common. I mean, it's very common in the sense that it's very abundant. It's very abundant because it happens in only a few plots. And there are many, many individuals in a few plots. So abundance and spatial distribution are independent, both in theory and to some degree independent in practice. These two species are among the most abundant species in the Amazonian forest of the Mariri area. Both are among the 50 most abundant species, which accumulate more than half of the individuals in this type of forest, in this region in particular. So from this second definition of commonness, some species are common because they have many individuals, other species are rare because they don't have many individuals. And these two aspects, two independent aspects of commonness can be combined in a single framework. Here I find this, and this is the definition of commonness that I like most and the definition that I'm going to use in the talk. Here we can distinguish species which are locally abundant but not very frequent. This is the frequency or spatial distribution, from species that are frequent, so they are common in one sense, but not very common in the other sense. And there are common species which are both frequent, they occur in many sites, and they are abundant in most of these sites. This is what I'm going to call common from now on. This two-dimensional framework for the study of commonness is potentially very useful, but it's not very easy to implement in practice. I will talk later about some ways of doing that. This two-dimensional framework is central to the oligarchy hypothesis. This is a very influential idea proposed 15 years ago by Pittman and colleagues. They reported that in tropical Amazonian forests in western Amazon, most of the trees, most of the individuals belong to a limited set of frequent and abundant species. They call these species oligarchic species. And an oligarchic species is something like this yellow star. And they reported also that these locally or regionally dominant species tend to be the same species in different regions across the Amazon. They reported the pattern for some regions, and they developed the ideas there, and they proposed some hypotheses that that in origin applied to neotropical forests, but can be applied in many other systems. I'm going to summarize a more general version of the oligarchy hypothesis that doesn't refer to this specific organism, not specifically for Amazonian trees, or not specifically for the scales that they employ for their study. And this general version of the oligarchy hypothesis has 3 predictions or 3 points. The first one is that in a region, most of the individuals belong to regional oligarchs. For example, these yellow and red species are both frequent and abundant in that region. And you can define the region at any scale you want. This could be 5 by 5 or 100 by 100 kilometers. The second prediction of this general version of the oligarchy hypothesis is that the regional oligarchs in one region will tend to overlap with the regional oligarchs of other regions. In this case, yellow and red species are oligarchs in both of the regions. And the scale depends on the study. So you can define region at any distance. The distance between both regions could be any scale of interest for you. The other species, uncommon species, it doesn't matter what happens with those. Could be shared between regions, could be completely different. So this aspect of the oligarchic pattern is fully compatible with high species turnover or high beta diversity. So species turnover and this type of oligarchic pattern are independent things. The third prediction, third point, is closely related with the second one, but includes also the uncommon species. And this prediction states that the abundance of the species tend to be correlated in the space. If you are abundant here, you will tend to be abundant in other regions. So these are the 3 main points of this general version, and many studies have addressed these points in one way or another. And most of the studies that tested these ideas accepted the oligarchy hypothesis in the sense that they found a limited set of frequent and abundant species, and these common species tended to be the same in different parts, different areas within their studied region. Importantly, a few studies conducted at a very large spatial scales or in highly heterogeneous landscapes rejected the oligarchy hypothesis. They couldn't find a limited set of common species, or these common species were not the same species in different places, or these common species were not so common compared with the average species. All these studies, or most of these studies, approach this idea from the yes or no perspective, from accept or reject the hypothesis. From my point of view, this previous body of literature suggests 2 things. First, that oligarchy patterns are common, are a general phenomenon in biological communities, but that can vary in strength from weak oligarchic patterns, where it is very difficult to identify which are the common species, to very strong oligarchic patterns, where it is immediately evident which are the common species in the region or in the study area. And because of the previous results, we can suspect that environmental heterogeneity and spatial scale play a role in placing different communities along this gradient. But we don't really know what are the drivers of oligarchic strength. And that is the motivation for the first study that I want to share with you, where we ask what are the drivers of the oligarchic strength. So environmental heterogeneity is expected to play a role. Heterogeneous environments would imply weaker oligarchic patterns. In the Amazon, the soil heterogeneity is it could be a major mechanism, but in other regions that encompass a broader elevational gradient, we expect also the elevational variability to influence the strength of the oligarchic pattern. If we have some common species at one elevation, other different common species at a different elevation, and we put everything together, then we have a weaker oligarchic pattern. The size of the study region per se can influence the strength of the oligarchic pattern. If the common species are aggregated in the space, imagine that we have a common species concentrated in a given area, and our studied region is more or less of the same size of that cluster of individuals, then that species is abundant and frequent all across our region. If that happens with all the common species, the pattern is a strong oligarchic pattern. But if we are covering a much larger area, that species that is concentrated around here is not frequent or is not very common in other places. If that is what other common species, if this pattern is general for the common species, then at larger extents the oligarchic pattern will be weaker. And this aggregation of kind of specific individuals of the common species could be completely unrelated to the environmental conditions, could be caused just by dispersal limitation. On the other hand, we expect stressful conditions to generate or facilitate stronger oligarchic patterns. If the habitat filtering is strong, there are fewer species that can be successful in those conditions, and those few species are expected to show stronger dominance in their communities. In our case, we expected stronger oligarchic patterns at higher elevations where the thermic conditions are more stressful. And we expected also stronger oligarchic patterns in more stressful soil conditions like hydromorphic soils, nutrients poor soils, or highly acidic soils, etc. A good analysis should take into account also important relationships between these explanatory factors. And this is the set of relationships that we evaluated in the Madrid region. To fit a model like this, it's not enough with a group of plots. We need many groups of plots. So we can have a group with the plots concentrated in a small area and a group of plots with the plots distributed in a large area. So we can cover the variability in the extent of the study area. The same applies to elevational variability. If we have many groups of plots, we can have a group of plots with the plots in a narrow elevational band, another group with the plots distributed in a broad elevational band. So we can, with many of these groups of plots, we can cover the whole variability in elevational variability, and so on. So to obtain these many groups of plots, we extracted groups of plots from our pool of plots. This is a plot network that we established in the Madrid region, which is close to Peru, northern Bolivia. There we have established 400 plots. For this study we use 400 plots, and these plots are distributed from the Amazonian forest, around 300 meters in elevation, this green area, to close to the tree line, around 4,000 meters in elevation. And with these 400 plots, we have estimated the abundance and the distribution of 2,500 woody plant species. So we have a pool of plots. We took 50 plots from this pool, evaluated all the factors. We took again 50 plots from this pool, evaluated all the factors. We took again. So we had, at the end, we had 500 subsets of 50 plots. The sampling was not at random and was with replacement. So we had some groups of plots in the small areas or homogeneous subregions, and groups of plots in larger areas, etc., and at different elevations, etc. Once we and measuring the elevational variability and the mean elevation, etc., is not very complicated. What is complicated is measuring the strength of the oligarchic pattern. How to combine frequency, the number of plots or the proportion of plots where a species occur, with the local abundance of that species in those plots. And we tried with two different methods. The first was the F-index of codominance. This index is the probability of taking two plots from the pool of plots from your study area, taking two plots at random, then taking two individuals from these two plots at random, and the probability of these two individuals of being con-specific. And this F-index can be high only when the species are abundant in many of the plots. It is low otherwise. The second method was the correlation between the local abundance and the frequency. For example, this region, this represents a region where the oligarchic pattern is weak, because the individuals don't belong to frequent and abundant species. Many individuals belong to species like this, abundant but not frequent. On the other hand, this community in the right shows a strong oligarchic pattern. Here, most of the individuals belong to species that are both abundant and frequent. And these two metrics, this correlation and the F-index, are correlated. So both are measuring or estimating the same phenomenon. Once we had everything measured at any of these subsets of plots or subregions of study, we fitted a path model analysis. With this model, each of the relationships receives an estimate of effect size, which is similar to the coefficient in a partial regression. And these are the results. Here, the black arrows are positive relationships, and the white arrows are negative relationships. And the thickness of the arrow is proportional to the strength of the relationship, to the effect size. The most important result here is that extent per se, once the effect of the environmental heterogeneity has taken apart, extent per se is the most important driver of oligarchic strength. So if we have two subregions with the same environmental conditions and with the same environmental heterogeneity, the subregion with the plots distributed in a larger area showed a weaker oligarchic pattern. Elevational variability is also an important driver of the oligarchic strength. So if we have two regions of the same size and at the same mean elevation and mean soil, same average soil conditions, if the plots are distributed in a broader elevational band, the oligarchic pattern was weaker. And interestingly, the effect of soil heterogeneity added almost nothing to the effect of geographical extent and elevational variability. And this was quite somewhat unexpected result, because the soil heterogeneity has been emphasized as a major driver of oligarchic strength and oligarchic patterns. So maybe in the Amazon, soil heterogeneity is important, but it doesn't. It's not the case for the whole elevational gradient that we evaluated. In general, this right side of the model is in agreement with our prior expectations. Large heterogeneous areas have weaker oligarchic patterns. The left side of the model is in disagreement with our prior expectations. We found a weaker oligarchic pattern, a slightly weaker oligarchic pattern at higher elevations. And we didn't find stronger oligarchic patterns in more stressful soil conditions. Here, elevation and soil are strongly correlated. So this is indicating that there is an elevational gradient in soil conditions and in oligarchic strength too, with weaker oligarchic patterns at higher elevations. And this is quite surprising, because previous studies suggested the opposite relationship, and this is also in disagreement with some things that are very evident when one is visiting different sites at different elevations in the Amazon and this transition. So this is the gradient that the model is reflecting. Strong oligarchies at low elevations, weak oligarchies at high elevations. And one possible explanation is that in the lowlands, if the common species in the different sites are the same species, at this multi-site scale, at this more broad scale, we will have a strong oligarchic pattern. And if the species, the common species here and the common species there are different, at that multi-site scale, multi-locality scale, the oligarchic pattern can be weaker at higher elevations. And this can be fully compatible with our prior expectation, if our prior expectation refers to a more local scale, if our prior expectation refers to single site scale, a single locality. And this gradient at this more local scale is true and is very clear in the field. In upper mountain forest, when you arrive to a site and you establish a camp there, and you establish plots around your camp, there are some species, 3, 5, 6 species, which are in all the plots, and then that are also very abundant in all the plots. And that pattern is very clear at high elevations, not so clear in the Amazon or in the lower mountain forest. So this is true. Both gradients can be true. Both gradients are compatible. What we have here is a shift in the direction of the gradient that depends on the scale. And that means that we have an interaction between extent and elevation. And we didn't include this interaction in this model. So from my point of view, this model was limited in describing properly the elevational gradient. And that is the motivation for a second study, where we try to understand better what is the elevational gradient in dominance patterns. In the second study, instead of mixing together many different spatial scales, we fixed the scale. We defined the scale for one site was always the same, and the distance between sites was always the same. And we asked if the abundances of the species were correlated between different sites or localities. And we had two hypotheses for this question. The first hypothesis emphasized the role of local processes in generating regional scale oligarchic patterns. So if the conditions here determine the abundances of the species here, the abundances of the species will be correlated as long as the conditions are the same. And the prediction along the elevational gradient for this hypothesis is that there is no elevational gradient in the correlation of abundances between sites, because, well, if this site is similar to this site, the abundances will be the same, and the same applies at higher elevations. So the prediction is lack of gradient, or lack of elevational gradient. The second hypothesis emphasizes the role of this connectivity between sites, regardless of the environmental conditions. And this hypothesis states that to have correlated abundances between different sites, we need connectivity between the different sites. If the different sites are disconnected, the abundances of the species will be different just by drift, stochastic drift in abundances. And the prediction for the elevation of this hypothesis is that in the Amazon or in the lower mountain forest, where there are no major barriers to dispersal and the connectivity between sites is easy, we have correlation of abundances between sites. But at high elevations, where there are major barriers to dispersal, deep valleys, mountain ridges separating different sites, we will have independence of the abundances between different sites. So the prediction for the elevation is a decrease in the correlation. And we evaluated these two hypotheses, or this question, at 4 elevational bands using sites separated 100 kilometers from each other. And these are the results. Each panel is one elevation. These are sorted from Amazonian forest to upper mountain forest. Each point is one species. The x-axis is the abundance, the relative abundance of the species in one site, and the y-axis is the abundance of the species in the other site at the same elevation. And the black diagonal is the null hypothesis that the abundances of the species are exactly the same in the two sites. And we found a deviation, we found a deviation from the diagonal that increases with elevation to the point that upper mountain forest, upper mountain sites above 2,800 meters in elevation, the species can be the same, but the abundances of the species are completely independent from each other. And this is in support of our second hypothesis, which emphasizes the role, the importance of landscape connectivity to keep the abundances correlated across the space. So the main conclusion from these two studies is that the regional connectivity and dispersal limitation is important and is apparently the most important driver of oligarchic patterns in this system, in the Andes Amazon transition. The second conclusion is that niche processes, deterministic processes, are of secondary importance. Maybe important in the Amazon, probably important in other systems like temperate forests, but in the Andes or or at higher elevation in these systems are not so important at dispersal limitation and connectivity. And this has important consequences. If we want to predict in a climate change scenario which species will be common in different places in the Andes, we are assuming that we can predict that based on a deterministic process. But these results indicate that to a large degree the species that are common in different sites are unrelated and are determined by stochasticity and abundance drift of abundances in the different sites. And a more general conclusion is that quantitative approaches to the oligarchy hypothesis are possible and are potentially powerful to describe floristic patterns in tropical forest. Now, that was the Madidi region part. All these results refer to the Madidi region. Now I will present some projects, some future directions at other scales or in other systems. As I said, I think there are interesting questions related to the study of commonness and also important applications of the study of commonness. I'm going to present 3 projects. 2 of these projects just started this last year, and this one I hope to move on during the next 1 or 2 years. The goal of the first project is to develop and test one hypothesis related to the origin of oligarchic species, on how these oligarchic species can be so common. The second project is related to monodominance in tropical forest. Monodominance is extreme case of dominance where only one species dominates, which is very unique pattern if you consider that tropical forests have thousands of species at local scales. And the third project is related to the elaboration of a high quality floristic map of the Amazonian trees, which we need and we don't have. So the goal of the first project is to develop and test the internal ecotypes hypothesis. Let me explain the motivation and the main prediction of the hypothesis. This is a tropical region, and these are 4 different habitats. In this region, we have different types of species. We may have a specialist species, which is successful in one habitat, but not really successful outside that habitat. And we can have also a generalist species, which is not really successful in any of the habitats, but can survive in different habitats. And both species are equally successful overall. So these 2 species fit into the generalist specialist trade-off. Besides, we have oligarchic species, and these species are abundant in many places, and they are occupying large areas, and they are as successful as habitat specialists in different habitats. And we don't know how these species can do that. They are ecological generalists, but they are not paying the cost of being a generalist. They have the advantages of the generalist species and the advantages of the specialist species. And what the internal ecotypes hypothesis tries to explain is how this works, how these species can do that. This is the main idea. The main idea is that there is substantial internal variability within the oligarchic species. There is differentiation into different ecotypes. And these units could be ecotypes, could be subspecies, or could be cryptic species within complexes of poorly resolved species. It doesn't really matter for the idea. The important thing here is that within oligarchic species we have different units, and all these units are more or less equally successful at, overall. So we would be explaining extraordinary success without the need to appeal to extraordinary success. And we just received some funds to test this idea in the Amazon. We will try to visit different locations and sample different populations of 10 or 12 common species, common tree species in the Amazon. We'll try to take samples for to evaluate the genetic structure and to evaluate if there are local adaptations, functional local adaptations. We'll take soil samples and functional traits from the different populations, and we'll try to identify if this is a general pattern or if this is something that doesn't apply. The second project is a collaboration with the German Center for Integrative Biodiversity Research, IDIF. It's a working group trying to explain monodominance in tropical forests. Classical monodominance. Classical monodominance is the coexistence in the same landscape of a mixed tropical forest with many, many species and patches of monodominated stands, where 60% or more of the individuals belong to a single species. And this is a classical monodominance referred to that that we don't these forest types are not distributed because of different soil conditions or fluid regimes or anything that is very obvious. So essentially we have radically different forest types and no clear explanation of why they coexist in the same landscape. I am most interested in the negative density dependence effects and its interaction with dispersal limitation to generate monodominated patches within mixed forest. I am going to work with permanent plot data from the Congo. There, the Center for Tropical Forest Science established four plots in the same locality. Two of the plots are in monodominated Gilbertiodendron stands, and the other two plots are in mixed forest. Altogether, these four plots contain 300,000 stems, and they have censused these plots several times since 1994. What is interesting here is that the species from the mixed forest occur also in the Gilbertiodendron forest. They can reproduce and grow there. And Gilbertiodendron appears also in the mixed matrix. So we have a pool of species. We can evaluate the dynamics of these species in two different forest types. Negative density dependence is the negative effect of the presence of conspecifics on your performance. So we can plot a metric of performance, like the probability of survival, against the density of conspecifics in your neighborhood, and the steepness of this slope is an estimate of the strength of the negative density dependence. And we can do this for many species. So we have many slopes, and we can estimate the distribution of these slopes on the community. We can do a similar thing with the dispersal potential, maybe using seed size data as a proxy for dispersal potential. And we can estimate the distribution of this trait for the whole community, for the whole pool of species. Gilbertiodendron, which is this monodominant species in this system, Gilbertiodendron, we know Gilbertiodendron is around here and around here in both of these distributions. Otherwise, it would be impossible to have such a strong aggregation of conspecifics. But there are two possibilities. One is that Gilbertiodendron is a radically different species. It's far from the distribution for the pool of species. It's an outlier. It's completely different in both aspects. So we will be explaining a radically different forest type because we have a radically different species. Then the question is, why don't we have species here if being here is so good? Another possibility is that Gilbertiodendron is not an outlier, is not an extraordinary species. It's a species that fits into the distribution of those traits for that that pool of species. But it happens to be the only one that is on the tail in these two distributions. And then find this. This would be a parsimonious explanation, because we don't need to appeal to a radically different species to explain the presence of two radically different and stable states of the same system. It's a little bit more complicated than that, but those are two alternatives that explain the presence of monodominated patches in very different ways. And the last project is the elaboration of a high-quality floristic map for the Amazon. From my point of view, it is impossible to do this map if we are trying to include all the floristic information that we have. At any given locality in tropical forests, most of the species are rare, and we fail to record the presence of rare species. So we know nothing about the distribution of rare species, but we know something about the distribution of a few common species. And I think we should take that information and exploit it. I think the moment to do this map is now, for two reasons. First, there are several initiatives to share data from plot data. There is ATDM for 1 hectare plots. There is a very recent compilation of a small plots data. There are compilations from gray literature that were not available a few years ago. And there are also some groups that work at the Amazon wide scale, focusing on certain taxa. And all these people are willing or interested in joining forces to do the map. Another important thing is that now we have freely available remote sensing products, or more easily accessible remote sensing products. And this is important because remote sensing products are unable to record the presence of rare species. They cannot give you a list of species or anything like that, but they may be good enough to help to detect shifts in the composition of the common canopy species. So the combination of these two things is what makes possible to do this map. To draw a map, or a floristic map, is basically assign each pixel in the Amazon to a floristic unit. And there are two approaches to do so. First one is take 300 or 500 species of the most common species and do a species distribution model for each of them. Then stack all those models together and estimate the relative abundance of these common species at each pixel. This would be enough to estimate the relative abundance of maybe 70 or 80% of the individuals at each pixel, and the other information we disregard. The remaining 30 or 20% of the individuals belong to rare species, and we know nothing about their distribution, and we won't know for decades or centuries. Once you have the relative abundance of these common species at each pixel, you can do a cluster analysis with the millions of pixels. The advantage of this approach is that you don't need much external information. You can even do these maps, these individual maps, without external information, just based on spatial structure between the observations. So you don't need remote sensing information to do this. The disadvantage is that you need a big computer to do a cluster analysis with millions of pixels. I mean, maybe there is not a big enough computer to do this. Second approach, instead of doing a cluster analysis of millions of pixels, takes a different way. We would take thousands of plots, and we would make a cluster analysis for those plots, which is a more tractable problem. And then we assign each plot to a floristic unit. And then with that information and the remote sensing information, we train the machine. We tell the machine, hey, this pixel is floristic unit 1. I know that because I have a plot there. I did the cluster analysis. And this pixel is floristic unit 2. I know that because I have a plot there. Now with that information, classify all the remaining pixels. And that is how a computer would do the map for you. And with that, I want to thank you for your attention, and I want to acknowledge the funding agencies that supported the work in the Mariri during 15 years and that are supporting my research now. Also the people that were involved in the field work and in the identification of the thousands of botanical specimens, and very especially to the researchers that established plots in the Mariri and shared the data with me to conduct this research. If you have comments or questions, I will be happy to address them.
Thank you. Questions?
So this is just a clarification. I couldn't understand what was the scale at which dispersal limitation was working. Was it regional or local?
The importance of the dispersal limitation is always regional, because the oligarchic pattern is something that is measured always at a regional scale between sites. And the first study covered sub-regions from 25 square kilometers to the 200 by 200 kilometers scale of the whole region. And the second study evaluated oligarchic patterns at the 100 kilometers scale distance. So the distance between the sites was 100 kilometers. And
So for the species that were top of the mountain, you were saying they are dispersal limited. The species was present at the top of the mountain.
yes,
So it's not dispersal between the mountains.
the species can appear in both sides, and then the dispersal can happen between different sites at that scale. And you can take, it may take many steps, intermediate steps, to connect the two sites. The idea is that the number of steps or the frequency of that dispersal is much lower at higher elevations. So the species, you can have a species here, and sometimes you have a dispersal event, and then you have 1,000 individuals here, and you may have 5 individuals there. But this dispersal is so infrequent that it's not strong enough to really mix the abundances of the species or maintain similar abundances across the entire region. So it's not that they are completely disconnected; they are less connected.
Could you tell us a bit more about how you would test for ecotypes and adaptation at those regions? You talked about genetic architecture, but I can imagine, you being able to demonstrate genetic architecture that really didn't have anything to do with adaptation to those local conditions. So how are you actually going to test to see whether there's...
Well, yeah, those are two things. We need to demonstrate that there are local adaptations, so we need to measure traits that correlate with the conditions, the local conditions, and we need to measure the conditions in a relevant way. So we may measure the soil conditions, and we may identify differences in soil, but if we are measuring the wrong trait, we will be looking at similar things. Then we cannot say that there are local adaptations. So that's the first step, and that's the first challenge. We will try to measure the set of traits that are commonly measured, that have like standard protocols to be measured, like specific leaf area, wood density, and so on. But we may fail on that part. Second step is if we can correlate the functions and the conditions, then is there a genetic structure underlying those things? And that is like the second thing, and we can fail also in doing that. So if we knew what are the relevant traits for the species to adapt to the environment, then we would measure those specific traits, but that is a big question, and it's not so clear which are the relevant traits.
And common gardens is just too difficult, takes too long.
It probably takes too long. Well, there are many ways of addressing this, and could be transplant experiments, but it's difficult to move plants between countries, so we should focus only on Brazil, for example. So it's all kinds of logistic complications.
I'm interested in going back to the very beginning with the definitions of oligarchy.
Uh-huh.
And you started by talking about how the two dimensions of commonness are independent of each other.
Mm-hmm.
And so I'm wondering if that's an alternative approach, because if they are independent, it's only a small subset of species that are both. And if you were to look at what are the traits or what are the conditions under which you're frequent versus conditions under which you're abundant, it might give a larger set of species and more variation to work with.
Yeah, it's true. For example, for this internal ecotypes hypothesis, we want to compare the widespread species that are frequent, that have different populations in different sites, but are not really abundant in those sites. So we need to compare these different types of species.
So are there ideas about what might be determining frequency versus what might be determining abundance?
Yeah, frequency is driven by niche breadth. This is a so simplistic explanation that I'm ashamed, but to be in different places, you need to have a broad niche. And the opposite applies to the local conditions. To be locally abundant, you need to be specialized in those conditions, and that usually means that you have a narrower niche, but because you are a specialist and you are abundant there because of that. But this is not really easy to do, because we have realized niches instead of fundamental niches, and these things are not even unimodal for the common species, are multimodal.
Yeah, that's what I was going to ask. Do we really know that those are the correlates or the ones? We always just assume that those are the correlates of those.
Mm-hmm. And also the niche breadth is such a, it means nothing. Niche breadth along what?
Any more questions? Any more back there? Robert?
Who's your ultimate degree work? Did you use small diameters of stems as well as large, so that you were capturing a picture of the full range of ages of the species, or was it limited to just large individuals? And with that number of individuals that make an only large?
You mean the Mariri network?
Yeah.
This network includes woody plants of 2.5 centimeters and above. It includes lianas, it includes trillets, shrubs, and also canopy trees.
Did you subdivide that to determine whether or not
No.
it was?
That's the goal of one project that is using information, is crossing data from between ATDN, which are big trees 10 centimeters and above, and this recently compiled network of smaller plots, and smaller plots usually focus on the smaller stems. So we want to compare what are the dominance and the rarity differences or similarities in small individuals. Could be individuals of different ages, or could be different species that are understory species instead of canopy species. We want to compare both things. Yeah.
All right, well, let's thank Gabriel again.
Yeah.